Let me introduce Dr. Meyer. David Meyer is the president and CEO of the LUNDQUIST Institute for Biomedical Innovation at Harbor UCLA Medical Center, formerly Los Angeles Biomed. After obtaining a Ph.D. in biology from UCLA, he spent 13 years in Europe, first as a researcher instructor in biochemistry at University of Basel in Switzerland, and then as a research group leader in cell biology and dean of graduate studies at European Molecular Biology Laboratory in Heidelberg, Germany. In 1987, he returned to join the faculty of UCLA School of Medicine in the Department of Biology, Chemistry. Aside from teaching and research, Dr. Meyer served as Associate Dean for the Basic Science and then as Senior Associate Dean for Graduate Studies at UCLA. Dr. Meyer research into molecular mechanism of membrane biogenesis and secretion has resulted in numerous peer-reviewed publications and reviews, including articles in Nature, Cell, and the Journal of Cell Biology. In 2005, he has appointed professor at UCLA. Prior to this arrival at LA Biomed in 2009, Dr. Meyer served as a vice president of research and scientific affairs of Cedars-Sinai Medical Center, and then as executive vice president for research at the House of Ear Institute. So as you see, he has a major background in chemical and administration of important institution of Los Angeles area. The reason we invited him is just to know what is new with medical science. And since we are almost neighbor, and my neighbors have helped this institution to fly even higher, we would love to hear from you, Dr. Meyer. And thank you, Matthew, for helping to arrange this meeting. Floor is yours, sir.
Thank you. Well, I would like to thank Susan Sharifian, who I know is on this call, for bringing up the whole idea of speaking today. And of course, Dr. Madani, to you as well for the organization. Yes, we are neighbors. I'm speaking to you right now from Lower Malaga Cove. So you can look down on me from where you live. And I look forward to telling you all about what's going on at the LUNDQUIST Institute, but give you a historical perspective as well. I hope what you will realize when I'm done is that even though you may not be familiar with the LUNDQUIST Institute or what we've done, you will see that in many ways, a lot of what we've accomplished over the years actually has touched your lives directly. And that's a tall statement, but I think I can back it up. So hopefully that's a good teaser for you to enjoy the talk. And I'll start with telling you a little bit about the history. Now, in 1943, this is what our campus looked like. We were the Los Angeles Port of Embarkation Station Hospital. This was built by the Army for the troops that were going out to the Pacific Ocean to fight in World War II, and for those who were lucky enough to come back. Now, in 1945, the war was over, and here was this functioning Army hospital. The County of Los Angeles at that time had one hospital. It was called County General Hospital, and I'm sure many of you are familiar with it. It's that big, tall building in Boyle Heights that's now been evacuated, if you will. And no one has a good idea what to do with it because County USC, which is the medical center, they built a new County General Hospital, which is now called the County USC Medical Center. In any case, that hospital, even though it had 1,000 beds, was overcrowded. And so the county was looking for more beds. And sure enough, this Port of Embarkation Station Hospital came on the market, and the county bought, believe it or not, 72 acres and all of these buildings for $46,000. And what did they do? They created Harbor General Hospital, which ultimately in 1978 became the Harbor UCLA Medical Center. But up until then, many of you probably remember it as Harbor General Hospital. So let me tell you a little bit about how this became an academic medical center. Now, what you have to know is that an academic medical center has three parts. It has to do clinical care, it has to do teaching, and it has to do research. Well, the clinical care was taken care of because there was Harbor General Hospital, and it started in 1946, and there were a lot of county physicians who either were hired or moved from County General Hospital. But then in 1945, the regents of the University of California decided, well, they have a campus in Northern California with a medical school. They have a campus in Southern California, UCLA, without a medical school. So they decided in 1945 to start a medical school, but UCLA had a serious problem. They had no hospital. They had no teaching faculty. They had no research facilities. And so in the late 40s, UCLA started its medical school on the campus of Harbor General Hospital. And in 1951, the first class of medical students was recruited. And up until the late 50s, when they finished building the Center for the Health Sciences at UCLA, UCLA Medical School took place in Torrance at Harbor General Hospital. Now you had two kinds of doctors. You had the doctors who were employed by the county, and you had the physicians who were the teachers on the medical school faculty at UCLA, all on the same campus. And they're missing something. They're missing the research. So those two groups of physicians got together, and in 1952, they started a not-for-profit independent research institute to administer all of the research going on that campus, serving both the physicians from the county and the physicians from UCLA. And as you notice by the date, next year will be our 70th anniversary. We've been around that long. So let me tell you a little bit about what's happened in those 70 years. What you'll first of all need to know are some facts. Like I said, we've been around for 69 years. We have over 100 internationally recognized research groups, everything from very basic research like figuring out the next vaccine against COVID to very clinical studies such as actually doing the clinical trials for the NIH for the vaccines and the monoclonal antibodies that were developed to treat or prevent COVID infections. And we cover every topic under the sun because these research groups are usually headed by physicians who see problems in their specialty, whether it's pediatrics or medicine or obstetrics or gynecology or psychiatry, whatever it happens to be. That's what they do their research on. At any given time, we have 600 research projects that are ongoing. Most of them are funded by the federal government, as well as by the Department of Defense or foundations or philanthropy. We publish a lot every year in peer-reviewed journals, and these articles are cited many, many times every year because they are that important. Now, we're also very entrepreneurial. What I mean by that is we don't just let our discoveries get published. We want our discoveries to become the next set of tools that physicians can use in the clinic, whether it's a diagnostic, whether it's a therapeutic, or whether it's a device. So we are very much interested in what we call technology transfer, and that's why we have filed numerous patent applications. We've had numerous patents issued. In the last 3 years, we've had 24 new patents issued on our discoveries. And in the last 12 years, which just corresponds to the time that I've been at the LUNDQUIST Institute, we've actually spun off 14 biotech companies that were based or are based on the technologies or the discoveries that were developed at the LUNDQUIST Institute on the Harbor UCLA campus. So now, let's talk a little bit about the accomplishments in the science. I would categorize them in 4 categories. Now, this should be over the last 69 years now, but we'll talk about the past 65 before we get into the present and the future. I would say the 4 categories would be new paradigms, new diagnostics, new therapies, and new devices. So I'll tell you about maybe 2 paradigms, a diagnostic, maybe about 4 therapies, and a new device that have been developed at the LUNDQUIST. And here's where I'm going to stick my neck out and say that every single one of you has in one way or another benefited from one of these. So let me start with a paradigm. And that is that in the late 60s, we had ambulance drivers and we had receiving hospitals. We had no first responders. And so the gentleman in the brown jacket, Michael Criley, who's now 90 years old this year, back in the late 60s, together with the County of Los Angeles, set up a training program to train county firemen to become first responders. Looking for a name for these first responders, they hit upon paramedics. Now, paramedic was an older term, but it referred to physicians who jumped out of airplanes in war zones. Parachute, medic, paramedic. But that was never applied to the people to whom it's applied today. Well, there was a television show on in the 70s called Emergency that ran from 1972 through 1979. They were all based on true stories from Harbor General Hospital. The episodes were filmed at the hospital or right down the street at the Carson Fire Station on Carson Boulevard, Carson Avenue. And lastly, that's how the term paramedics became popular throughout the United States. The idea came from the Lundquist Institute, and the name was adopted and adapted by everyone in the United States who now refers to these people as paramedics. These are hospital-trained people. It's a grade above EMTs. So it's the top level of first responders. So that's a paradigm that we came up with at the Lundquist Institute. I'm sure many of you have dealt in one way or another with paramedics in LA County, which you would not have done without us. Here's another one. We had a women's healthcare center treating women who were from underserved and underrepresented minorities, which is, of course, the community that we live in to a large extent. And this was offering free medical care, and a lot of it had to do with tests. It had to do with advice. It had to do with giving tests for sexually transmitted diseases. It had to do with doing pap smears. And the chair of obstetrics and gynecology at Harbor realized that this was something that you don't need to be board certified in obstetrics and gynecology to carry out. And there were these nurses that were handing the instruments to the physicians to do the tests and taking notes on the outcome. Well, we determined that these people, they were all women at the time, could actually do the tests. And so we came up with the concept of nurse practitioners, particularly in women's health. We trained the first 2,000 nurse practitioners in the United States and were awarded the first government support to train them. And they went out to 14 different states of the United States, Western states, where there were no physicians at the time, to serve the underserved. Now, of course, nurse practitioners is a common profession. It's taught in nursing schools, it's taught in professional schools, and I'll bet that most women nowadays, if they go to see their gynecologist, end up seeing a nurse practitioner, at least at the outset. And many men now find themselves in the hands of nurse practitioners when they go to the doctor's office. Another pioneering paradigm from the Lundquist Institute. Let me tell you a little bit about some of our therapies. Now, I'm going to focus on therapies, by and large, but not all, that have saved the lives of children who were born with inherited fatal diseases or congenital problems, either in utero or at birth. So let me start by talking about this fellow here. Now, he has an eye infection and could very well go blind because his mother had a sexually transmitted disease, which transferred to his eyes just by being born. Well, in the old days, we used to put drops of silver nitrate, you may remember this, you physicians, particularly pediatricians, into the eyes of newborns with the idea of it being antiseptic and killing any pathogens that might infect the eyes and even lead to blindness. Well, this fellow, Sherwin Eisenberg, who is an investigator at the LUNDQUIST was the first pediatrician ever to be board certified as well in ophthalmology. And he traveled to Africa and was appalled by the number of kids who were going blind because of these congenitally acquired infections from their mother. And so what he decided to do was first to test silver nitrate in a clinical trial. It had been grandfathered, it had been around so long, it was there before the FDA. So just like aspirin, it never had an FDA trial, and everyone believed it was good and it was being used. He did a clinical trial and found out it wasn't all that good. And it was so expensive and couldn't be used in places like sub-Saharan Africa. So Dr. Eisenberg came up with low-cost iodine-based eye drops. They are so cheap, you can treat 5 babies for a penny. $17 will buy you a gallon. And as a result, just in Uganda and other parts of sub-Saharan Africa, these eye drops have been used in tens of millions of babies already. And if you do the math, you can easily calculate that the number of babies that would have gone blind, which didn't go blind, amounted to 500,000. Just by these low-cost eye drops, they don't require refrigeration. Anywhere where there's a newborn, anywhere where there's an eyedropper, and some of these eye drops, you can put them in the eyes of the babies and they will not get the infection and they will not go blind. So there's an example of discovery becoming a therapy. Let me tell you about another one. This is a diagnostic. This is the test for serum cholesterol. Now, I'm willing to bet that everyone here knows what their serum cholesterol level is, and whether it's too high and you're taking statins, whether it's low enough that you don't have to, whether you've changed your diet, whatever it happens to be, you can thank us that you know that, because we pioneered the test for serum cholesterol for total cholesterol that's still in effect today. Here's another one, and that is surfactants. Now, many of you know, particularly pediatricians, are aware of the fact that if a baby is born prematurely, particularly about 5 weeks prematurely, there's about a 50% chance they will die. At least that was the case. But it was determined that the reason they were dying was something called infant respiratory distress syndrome. Well, what causes that? What causes it is the mere fact that your lungs are not developed until just before birth. Why? Because you don't need them. You have a placenta, you're happily living off your mother, and you're not breathing. But come time to be born, you need to have fully developed lungs, and that means that these lungs have to be secreting a chemical called surfactant. And for those babies that are born prematurely, very often they're not. They have lungs that deteriorate very quickly. They have infant respiratory distress syndrome, and in many cases, they will die. In the 60s, Jackie Kennedy, the wife of the president at the time, gave birth to a son who died at 2 days of age. The reason? He had infant respiratory distress syndrome, and he was born 5 weeks prematurely. That was why he had infant respiratory distress syndrome. There were no surfactants at the time, and he's now buried next to his father in the Arlington National Cemetery. One of our physicians, a pediatrician, Alan Jobe, a neonatologist, came up with the idea of isolating substitute surfactants, and he started out doing it from pigs. And a very tricky technique is actually to use a tube and a hypodermic syringe and apply in these tiny babies surfactant directly into the lungs, and lo and behold, it was sufficient to enable them to survive. Now it's routine, and you have babies that are born 12 weeks prematurely, and they survive. Now, if 1 in 9 babies are born prematurely, which is true, and a large percentage of them, probably the majority, depending on how premature they are given surfactant, you can figure that there are tens of millions of adults who were born prematurely, who received surfactant and are alive today, otherwise would have died of infant respiratory distress syndrome. Again, another breakthrough discovery from the Lundquist Institute. Next, I want to talk about a genetic disease. Some of you may be familiar with mucopolysaccharidosis. It is an inherited recessive disease, recessive gene, which encodes a very important enzyme in the body. If you don't have it, you don't break down accumulated byproducts and garbage in your cells. It is an enzyme that these kids don't have. It's either mutated and not functioning, or they don't have the right gene for it. The problem is that death occurs by 10 years old, because the garbage accumulates in the cells, and when the cells accumulate it, they die. When the cells die, the tissues die. When the tissues die, the organs die. And when the organs die, the baby dies, or the child. It starts out with symptoms probably 2 to 3 years old, progresses through paralysis, ultimately blindness, loss of memory, loss of mental capacity, and by age 10, death. Well, Emil Kakkis, an MD Ph.D. from UCLA, decided that he could, because the gene that was actually defective in these babies had been cloned, that he could actually produce from this gene enough enzyme to transfuse that into the sufferers of mucopolysaccharidosis and reverse the effects by giving them the enzyme they need to break down the garbage. And this is called enzyme replacement therapy. And it is a product that's now available called Aldurazyme. In 1999, the first patient on a clinical trial of Aldurazyme was flown in from Texas, was treated at Harbor UCLA Medical Center on this new therapy. Within 2 weeks, he was already feeling better and recovering. And this was in 1999. His patient is Ryan Dant. And today, Ryan is 31 years old. He just got married this past summer. He graduated high school, which he never thought he would be able to. He continued to play baseball, which he was doing before the symptoms set in. He went to the University of Louisville, majored in sports management, had an internship with the San Francisco 49ers. I just met with him 2 weeks ago. He was on a trip and came to LA with his new wife. And this is another example of a discovery leading to a therapy. It's what Dr. Kakkis saw in his patient population as a pediatrician and decided, this gene is cloned and I can do something. And he did. And now there are thousands of adults around the world who are on enzyme replacement therapy. Ryan needs an infusion, an injection once a week. But as you can see, it's 21 years past the time he should have died. The next I want to talk about is another inherited disease, which you may be familiar with. It's called sickle cell disease. And if you look at this rendering here, you can see normal red blood cells. Those are the 3 that look like jelly donuts. And you have the sickle cells. Those sickle cells are stiff, they're sticky, and they clog up the finest capillaries, veins, and arteries in your body with disastrous results. It's a single gene deficiency in the globin part of hemoglobin, and it has disastrous consequences such that, for example, if you have sickle cell disease, you develop pain that's worse than terminal cancer. It's a pain crisis. You are hospitalized to treat the pain. While you're in the hospital, you may be getting blood transfusions to replace your defective cells with someone else's good cells. And because your circulatory system is so screwed up, your immune system suffers as a result, and you're on antibiotics, very often to prevent the most minor of infections as far as other people are concerned, but major as far as sufferers of sickle cell disease are concerned. These kids, largely kids, are in the hospital 6 months out of the year for visits, taking care of pain crises, blood transfusions, and antibiotic treatments. Now, as you can see, this kid is African American. There are 100,000 sufferers of sickle cell disease in the United States, and they all received the gene from slave ancestors because the genes came to the Western Hemisphere from sub-Saharan Africa during the slave trade. Now, this is very interesting. The reason that there are only 100,000 in the United States is that we're no longer in the tropics. Yet where there are tropical regions, there are 25 million people who have sickle cell disease throughout the world. And the reason is very simple. Those odd-shaped sickle cells are resistant to malaria. The malaria parasite, which needs red blood cells to reproduce, can't do it in sickle cells. So this is kind of an irony that you have one life-shortening disease to prevent you from getting a fatal disease. But this is Darwinism in its finest, it's natural selection, and that's why there's still 25 million people running around with sickle cell disease because they haven't been killed off by malaria. So it's a real problem. So Yutaka Nihara, one of our hematologists who saw a lot of sickle cell patients in our hospital, decided he had to do something to turn sickle cells back into normal cells. And sure enough, what he found is something that's now sold as Indari. It is so simple, it's mind-boggling. It's just L-glutamine. And I say just L-glutamine because that is a commonly occurring chemical in your body. It's the most commonly occurring free amino acid in your body. And if you make it into a powder and you stir it in water twice a day, those odd-shaped cells will suddenly spring back to a more normal shape, which he discovered in the laboratory, and then tried it on patients. And sure enough, these people no longer have pain crises. They no longer need transfusions. They're no longer on antibiotics. It's about 75 to 80% effective in treating all of the symptoms. So instead of being 6 times a year in the hospital, maybe they'll be once a year in the hospital, and then it's not nearly so serious. It has changed the lives of sickle cell sufferers in the United States. As I said, this is such a commonly occurring molecule that it was the first new treatment for sickle cell disease in 20 years that was approved by the FDA. But because it's so gentle and naturally occurring and 0 side effects, it's the first that was ever approved for use in kids, for pediatric use. Another example, discovery becoming a therapy. So that's sickle cell disease. Let me move on now to something affecting adults as well. Now, I am going to stick my neck out, but I'm very confident that the third leading cause of death, forget about COVID, this is pre-COVID, the third leading cause of death in the United States are hospital-acquired infections. The numbers that are reported already show you that it's the third leading cause of death behind heart disease and cancer. It's jumped over stroke. It's jumped over other COPD, for example, that used to be in third place. It's now in third place, and it's probably could even be higher because it's poorly reported. Hospitals don't want you to know that they have a problem with infections. So if someone dies of a hospital-acquired infection that they had only when they came into the hospital, the death certificate says died from complications of whatever they came into the hospital for. So if they had a kidney transplant, got an infection and died, the death certificate says died of complications of a kidney transplant. So it's masked. And you'll see in a minute that sometimes it's really poorly masked and even more dangerous. So what do you do against a hospital-acquired infection? Now, many of you are thinking, oh yeah, sure, I've heard of these staph infections and that must be it. No, the real culprit are fungi, molds. One of them is Candida albicans, and we're covered with Candida. And for most of us, it's inconsequential because we're not immunocompromised. So for people who have HIV infections, or people who have just gotten a transplant, or now people who have COVID, it's particularly dangerous because they are immunosuppressed. Candida loves to grow on plastic. And if you've just had a heart transplant, lung transplant, kidney transplant, you have tubes, plastic tubes coming out of you, going into you, and they're covered with Candida. And they're getting into your bloodstream. And guess what? We put you on immunosuppressive drugs so you don't reject the kidney. What we've just done is made you a sitting duck for a fungal infection because you can't do anything about it. And 50% of the people who get it in their bloodstream will die. So what do you do? And guess what? As I'll show you in a minute, many of the antifungal drugs don't work. And so just as we're developing vaccines for COVID, the idea is to develop vaccines against Candida. And that's exactly what Jack Edwards did over the last 20 years at the LUNDQUIST Institute. And this vaccine against Candida has already completed a phase 2 clinical trial. And that is just the start, because it gets much scarier. And that is something called Candida auris. That is another species of Candida, and it's now ranked as a superbug. A superbug is a pathogen that is virtually completely drug resistant. We deal with a lot of antibiotic resistant bacteria. Well, these are anti-fungicide resistant fungi. And it's so scary that this was a headline in the New York Times, and that there was a mysterious infection spanning the globe in a climate of secrecy and the rise of Candida auris, drug-resistant germ. It started out in London in a climate of secrecy because the hospital there didn't want the word to get out that they had an incurable fungal infection looming in their hospital. But it did get out, and it got to New York, and it got to New Jersey, and it got to Chicago, and it got to Orange County, and it got to Los Angeles, and now it is on the rise. It is hospital acquired, and there's very little that people have been able to do about it. And I wouldn't be telling you about it if we weren't already doing something about it. And for the last couple of years, we have been doing preclinical studies, which means treating mice, basically, who have Candida auris, or who we want to prevent from getting Candida auris with our anti-Candida vaccine. And you can see here, we have already published papers, and Dr. Ashraf Ibrahim is the lead researcher on this, and you'll see in a minute on other studies as well. And the title is that the fungal infections, as you can see, this vaccine, which is the vaccine against Candida, protects mice from multi-drug resistant Candida auris infection. They will be dead within days, the mice, unless they've been immunized, in which case they survive the infection. Now what has to happen is this has to shift into human trials, and then we can immunize people in advance against these hospital-acquired infections. Now it's going to get ugly, because there is something called the black fungus. The black fungus is something that is, and you can see these are headlines, Scientific American, the BBC, The Guardian. I could have gone online and given you dozens of headlines from various publications talking about what's happening in India that very few people know about. And that is that there is this thing called the black fungus, which is mucorales or mucor, and it causes something called mucormycosis. It used to infect a very small number of people who were immunocompromised, just like Candida albicans or Candida auris. But this mucorales, you can see here, is a picture of the fungus, and those little brown balls that are coming out are spores. These spores get into your lungs. 80% of the people with those spores in their lungs will develop mucormycosis and die. The other place it lodges is into your sinuses, and then it will progressively eat away your face and your brain, starting with an infection in your sinuses. And if you've just had dinner, you may not want to look at what I'm about to show you, but this is why it's called the black fungus. And this is what happens when it gets into your sinuses. And it looks like that. And there's nothing you can do for these kids or anyone who gets it into their sinuses other than very disfiguring surgery, because up until recently, there's nothing you can do about the black fungus. Now, what is astounding is that there was less than 10 cases of the black fungus, of mucormycosis, in one of the most populous states in India before COVID. And then as a result of a COVID infection and this horrible Delta variant that has completely taken over India, it went from about 6 in a year to 50,000 in a month of cases of black fungus, because COVID-19 patients are very often immunocompromised as a result of the COVID infection, and this has taken over. So again, this is due to the black fungus. Now, I wouldn't be telling you this if we weren't way ahead of the game. So once again, I'm going to tell you about Ashraf Ibrahim, who started a biotech company, and it's the first antibody targeting mucormycosis. Now I'm going to give you a little bit of a biology lesson and show you a little bit of data. The biology lesson is about monoclonal antibodies. Now, many of you have probably heard about monoclonal antibodies or already know about monoclonal antibodies, but they are basically an antibody that we can actually give to people as opposed to a vaccine. If we give you a vaccine, we're hoping that your body will produce an antibody. But what if you're immunocompromised? We could give you all the vaccine in the world and you wouldn't do anything with it because you don't have an immune system that's functioning. But how about if we just gave you the antibody directly? Well, we'd have to make the antibody. And when we give someone an already made antibody, that's called giving them passive immunity, as opposed to active immunity, which is what happens when we give you a vaccine. Active because your immune system has to be active to make the vaccine. So monoclonal antibodies are very straightforward, and someone won a Nobel Prize in the 80s for coming up with the idea. What you do is you take a mouse and you infect them with something against which you want them to make an antibody. So in this case, you give them mucor, you give them the pathogen, and they make antibodies. Now the problem is the mouse is making antibodies against everything the mouse comes into contact with, not just against mucor. So you have to identify those specific cells in the mouse that are making that antibody that you're interested in, the one against mucormycosis. So what you do is you actually isolate and screen all the cells from the mouse, and you identify and then clone, monoclonal, you clone those cells that make the antibody you're interested in. And those cells will grow in culture and produce boatloads of exactly the antibody you want. But there's a problem. It's a mouse antibody. And if I put it into a person, they're likely to reject it. Maybe not quickly, but they will reject it as foreign protein. So what you have to do is you have to make a human version of that mouse antibody. Well, we can't start using humans as mice. So what we do is we take the DNA from the cells of the mouse that are making that antibody, and we cut and paste. We clip out molecularly those regions of the DNA that encode the mouse parts, and we replace them with human antibody parts. And then the mice make what we call humanized monoclonal antibodies. There are a lot of them on the market for a lot of things. Any of you familiar with Humira, for example, that is a humanized monoclonal antibody. Just in case you're watching CNN from time to time and you want to know which drugs are actually antibodies, their technical name ends in AB. And if you see a drug that ends in AB, that means it's an antibody and is going to be used as a passive immunotherapy. So here's a result, the only data I'm going to show you. So here's how the study works. You start out and you give the mice a mucor infection. So they develop mucormycosis. You wait a couple of days, they have a full-blown infection. If you don't give them anything within 13 days, the red curve, every mouse is dead. This is a survival curve. 0 mice, all the mice surviving. If you give them the most commonly used antifungal drug, you'll give them an extra day of life before they all die. But if you now give them the humanized monoclonal, don't forget, you're now giving this into a mouse, so they're going to see that as foreign protein. Even if they see it as foreign protein, you can still save the lives of 20% of the mice. If you give the mice the mouse antibody and combine it with the antifungal drug that doesn't work, you save every single mouse. So now the real question is, what happens if you take the humanized antibody, combine it with the antifungal drug? In the mouse system, you save 70% of the mice. So now we are looking for someone to give us about $3 to $4 million, because that's what it's going to cost, to make enough of this humanized monoclonal antibody to do the trials that will get us all the way through FDA approval. This isn't cheap. It's very expensive, and that's why one has to find donors. One has to find supporters of the research to be able to fund it, because the federal government isn't in a position to give us an NIH grant that'll get us enough money in a short enough time to get this made, to get it through clinical trials anytime soon. So that's one of our problems in terms of doing the research at the LUNDQUIST Institute, is coming up with the money to bring it to the final stage, to get it to the patients. So there's your biology lesson for the day. Let me now move on to the last thing I want to talk about, which is a device. Now, some of you may have heard of long QT syndrome. Long QT syndrome, here's an electrocardiogram. It's one of 15 different genetic mutations of ion channels in the heart. Normally, the interval, this QT interval should only be about this long, the interval between this peak and this valley. Now, if it's longer, it's indicative of a problem. And what it's called then is long QT syndrome. So what about long QT syndrome? First of all, it affects 1 in 2,000 newborns. It's responsible for 10% of the sudden infant death syndrome cases in the country if it's not diagnosed. What that means is literally the #1 cause of death among newborns all the way up to 1 year old. Sudden infant death syndrome could be localized and treated if one were able to diagnose it. If you can diagnose it, you can treat it with chemicals, with drugs, very easily. It's 90% effective. And for those to whom it's ineffective, you can implant a defibrillator and they'll be fine and their long QT syndrome will be taken care of. Here's the problem. You have to do an electrocardiogram on a newborn because death will occur within the first year. So you've got to get to them when they're born to do an electrocardiogram. Now, if you look at a 3-month-old trying to get an electrocardiogram, all of us who've had electrocardiograms know that this is a major pain. You have 12 different leads that have to be hooked up to different parts of the body. And that's not enough because it has to be hooked up to the big box. And the big box has to have all those leads connected to it. Now, hospitals are not going to do this on every newborn. Pediatricians are not, you shouldn't wait until the first pediatric exam. You should do it at birth. So how can you do this? Well, Ray Chang, a pediatric cardiologist, came up with an ECG device that comes in an envelope. What do you get in the envelope? Which you can take home at birth and you can do an electrocardiogram on your newborn in the home. You don't need a nurse, you don't need a doctor. You can just take what's in the envelope, which is a disposable strip. You see it right here. That has all 12 leads. There's one up here, there's one down here, and there's 10 of them in this strip. So that replaces all those wires. So what replaces the big box? Well, this little guy here, that will, within 10 seconds of your pushing the start button, will generate a complete electrocardiogram, which will then, by Wi-Fi, be sent immediately to the physicians at QT Medical, which is one of our startup companies started by Ray Chang, and you will know in almost real time that your baby's okay. You send back the little box to us because there's a self-addressed stamped envelope, and you throw this out because it's so cheap to make it. Does it work? Well, I'll tell you that it received FDA approval about 3 years ago, 3.5 years ago, and it's now on the market. So how do we know it works? Well, the good news is that the NIH funded a clinical trial, and we went to the St. Francis Hospital here in LA, and we went home with 2,500 newborns and their parents with the envelope containing the micro ECG machine. And sure enough, we found one with long QT syndrome. Remember, 1 in 2,000? Well, if you do 2,500, you stand a good chance of finding the one. We also found 11 more that had to be referred for genetic counseling. We found 3 with electrophysiological defects. We found 3 with congenital heart defects, like a hole in the heart or a damaged valve. So just by doing this, we saved 18 lives. These are kids who would have died in their first year of birth had they not had an ECG as a newborn. This is something that has to be done for every newborn to save those lives. So what's the next step? Well, that strip comes in 7 different sizes. We have them now for adults. So one of the first places it's being used is on airplanes. Think about it. How many times have you heard stories about someone's having chest pains and the pilot has to land the plane and everybody has to get off and they have to call the ambulance? Well, now you can do that in the air, and everybody has Wi-Fi in the air, and you can find out whether it's just innocuous chest pains or does the pilot really have to land the plane in a hurry. But you can imagine it in ambulances. And the next study we're going to do is on high school athletes. High school athletes very often have sudden cardiac arrest or even sudden cardiac death on the playing field. Well, if they could have had an ECG before they started doing the sports, we would have known you shouldn't be doing this sport because you have a heart problem that needs to be treated first. And this is the next step, but you can imagine other places it could be used. Cruise ships, people who just had open heart surgery are at home. They now have chest pains. They don't know whether to call 911. Send this home with them. They slap it on, and within minutes, they'll know whether they have to go back to the hospital or whether they're just having pains as a result of post-surgical trauma or incisions or whatever it happens to be. Okay, so now you've seen what we've done. I've given you a sampling, couple of paradigms, a diagnostic, a few treatments, and a device. So let's now talk or shift to the present, and let's talk about the future. So what's the future? Precision medicine. You've heard this before. It's also called personalized medicine. We have an Institute for Translational Genomics at the LUNDQUIST. Now, let me start out by asking you some questions, and then you'll understand precision medicine a little better. So let's talk about medical disparities. Why do Hispanic women have higher rates of type 2 diabetes than other ethnicities? I'm not going to ask you to answer the question yet. Why do African-American men have higher rates and more severe cases of glaucoma? Why do Asians have higher rates of stroke? Why do Caucasians have higher rates of Alzheimer's? Well, what's the difference between all of these? It's what genes they have. So it's their genetics. Let me go one step further. How about this? You've heard this from smokers. Well, I'm not worried about that I smoke cigarettes because my grandfather lived to be 103 and smoked 3 packs a day. But you also hear cases of someone who died at 40 of lung cancer and never smoked a day in his life. Well, what's the difference between the two? Well, I dare say that the guy who lived to be 103 had one of two situations or both. He either had genes that prevented him from getting cancer or didn't have genes that promoted him getting cancer, or both. And the poor guy that died at 40 had the opposite. He either didn't have any genes that prevented him from getting cancer, which we know exist, and did have genes that promoted his getting cancer. So the difference between the two is exactly the same. It resides in your genetics. So in order to understand your genetics, you really have to be able, to sequence your DNA. So how difficult is that? Well, back in 1990, when they were doing the first human genome project and sequencing the human genome, this one lab set up with chemistry, set up with computers, set up with robots, was needed just to do one chromosome out of the 23 pairs that you have in your genome. By the beginning of this century or this millennium, you could do it on your desktop. Well, where are we in 2021? This is where we are in 2021. And yes, the device on the left is a laptop, and the device on the right is actually the sequencing machine. But if you think it stops there, no, it doesn't. It gets to this, which hooks up to that, and you recognize that as an iPhone in this case. There's a little well, I don't know if you can see it right here, a little hole, and you can put a drop of blood or a drop of saliva in there. It does all the chemistry through microtechnology, microfluidics, microchemistry. And the cell phone is just there to transmit the data to the cloud where someone can actually do the analysis of what's going on. Now, what does that mean going forward? That means that your doctor will have something like that, take a drop of blood, put it in there, and then get the results and be able to say, Harold, you're now a healthy 22-year-old. The genetic analysis from the saliva sample that you gave us last week tells us that by the time you're 60, you're going to have an 80% chance of developing prostate cancer. But if you begin this treatment today, your risk at age of 60 will drop to 5%. That's personalized medicine. It's based on Harold's genome and Harold's genome alone, based on what genes he has that are going to promote prostate cancer or what genes he has that are going to prevent prostate cancer. All you have to do is figure out what are the genes. And that's what we're doing at the Lundquist Institute. As a matter of fact, precision medicine is predictive because we told you 40 years in advance, we told Harold what's going to happen. We told him that we have a therapy and it was tailored to him specifically, right treatment for the right person at the right time. But as I said, 50% or probably a lot more of your likelihood of suffering a common disease resides in your genes. You can't just say it's based on your smoking cigarettes. Your genes will determine the outcome. And so at the Lundquist Institute, I'm pleased to say that these are all of the diseases whose genes we are studying right now, either on site or in collaboration with other institutes around the country or around the world. Cardiovascular, metabolic, neurological, vision, renal, and others. And you recognize many, many of them. All of these are what we call common diseases or complex diseases because it's not a single gene that causes it, like sickle cell disease or mucopolysaccharidosis. It's a combination of genes, combinations that either prevent or promote a particular outcome. We have to understand these. And once we do, and we have enough information, we will be able to assign you a polygenic risk score. What does that mean? Let's just imagine that all those little people there in this figure, we're analyzing them for whether they have heart attacks or not. And the ones in the green, very, very low chance of getting heart attacks. And we know what their genes are. And those people on the right, they've had multiple heart attacks and we know what their genes are. And if we have enough people in this study, we are going to find out where you fall on this curve. And if your genes are like this guy here, you're going to have a very high probability of suffering from a heart attack. But if your genes are more like this guy over here in green, you don't have to worry about it. There's no chance you're going to end up with a heart attack. So this assigns you a score. So let's say you happen to be right here, and we'll tell you, look, you have a 3.7-fold higher chance of getting a heart attack than the average person, or compared to 1 million other people in this study. That is what we can do already for cardiovascular disease and for certain kinds of cancers. And the more research that we do, the more people that we study with disease and know what their genes happen to be and how those genes correlate with the disease, we'll be able to tell you long in advance. We'll be able to tell you the day that you're born what you're most likely to end up with later in life. Think of how much medical costs that would save. Think of how much anguish you would have discovering all of a sudden that you have cancer, rather than knowing I am a candidate for cancer and I better go on these drugs or change my lifestyle. Or if you're a candidate for heart disease, I better get more exercise, start taking aspirin, take a statin, change my diet. All of the things you can do to prevent the outcome that your genes have already determined for you. But in order to do this, we have to continue to do the kind of research on these diseases at the Lundquist in our Institute for Genomics that we talk about. So the last thing I want to talk about in terms of science here is a couple more examples of the future. Here's a big one, literally, and that is a problem. And, you know, in the 1970s and 80s, maybe 20% of the population, no, there were no states in the United States where there was greater than 20% of the population being obese. Now, most of the states in the United States are between 30 and 40% rates of obesity. I'm not talking overweight. That's well above 60%. And as far as kids are concerned, right now, we have a 19% rate of childhood obesity and a 35% rate of kids being overweight. We've got to do something about it because it leads to all kinds of problems, particularly things like metabolic disease and diabetes. So we are starting a Center for Precision Nutrition, just like we have precision medicine. Why? Simply because each person, based on their genes and other things, will respond differently to specific foods and nutrients. Due to your genes, your environment, your lifestyle, and the bacteria that are growing in your gut. I'm not even going to get into the microbiome, but I'm happy to answer questions about it after my talk if you're interested in talking about gut bacteria and what effects that has. But the best diet for one individual is going to look very different from the best diet from another, but no one has ever done the research. We have these pyramids, right, of what you should be eating. Well, they're wrong, and people still use them. And they're not tailored to the individual. Just like there is precision medicine, there's going to be precision nutrition. Another center that we're worried about is Center for Vaccine Innovation. And God knows that's such a topical subject right now, particularly with COVID. But look at this. After billions of dollars and work by research institutes all over the country and universities, here is a list of very common pathogens about which we can do nothing because we don't have the tools. We don't have the vaccines to cure AIDS or influenza. You can just prevent it from year to year, but you can't take care of it. You can't immunize people. All of the fungi, which are in purple there, then all of those bacteria, including E. coli. You hear about the antibiotic-resistant E. coli killing people all the time. It's in the hamburgers, it's on the strawberries, it's in the lettuce, wherever it happens to be. Tuberculosis, same deal. And all of the parasites, Plasmodium, things that cause, for example, malaria and the like. So we're proposing, and we're putting together the funding for a Center for Vaccine Innovation, and we will have 4 research units focusing on fungal pathogens and vaccines, bacterial as well as viral, but importantly, a vaccine clinical trials unit. During COVID, we did 8 federally and drug company sponsored clinical trials for COVID. We are uniquely set up for doing vaccine trials. So why not open up a center where anyone who's developing vaccines anywhere in the country or anywhere in the world can come and actually do the clinical trials of their vaccine. And that's another thing that we're going to be doing through a Center for Vaccine Innovation in the future. So I want to finish up with the vision for the campus. Now, what I want to do, and what I've been striving to do for a number of years, is creating what we call a bioscience hub in LA County. Now, what does that mean? Well, I told you that our physicians start out at the bedside and they see a problem. They then go to their laboratory, better known as the bench, and that's where, for example, for sickle cell disease, they developed Endari. But then what we have to do is take that discovery and protect it with patents, which it then becomes intellectual property that we can sell or license, either to our own startup companies or to other companies. Now, the problem with startup companies is the following. In bioscience, most of the companies that are in business after 5 years were in an incubator, a bioscience incubator, during a very critical part of their development. That is doing the proof of concept, proof of principle, and attracting venture capital. And that's best done in an incubator. And so the next step would be to be in an incubator. That's great. Now you have funding, now you have proof of principle, now you've got to make this stuff in enough of a quantity to go back to the bedside and do a clinical trial. And for that, you need a manufacturing facility or a bioscience tech park. I can tell you there are 13 incubators in LA County, and I'll tell you in a minute that we have the best one. But we're working now to establish the first tech park for bioscience in all of LA County. And we have already been granted an option for the land on the campus, which I'll show you in a second. So this would be the vision, to be able to start at the bedside and go all the way back to the bedside through all of these steps and never leave the 72 acres that was once Harbor General Hospital. And that would be the first bioscience hub in LA County. And so where's the incubator? Well, this is our newest building. Please come by and visit. I'll give you a tour. The 3rd floor is 18,000 square feet of bioscience incubator. We currently have 35 companies, startup companies that are in there. A few of them are from us. A lot of them are from UCLA and USC. We have some from Korea, from Australia, and even from Argentina. There are 33, it's now 35 companies since I made this slide. This is what the incubator looks like. There's core equipment that was funded by a grant from the federal government. It rents by the bench. There are 4 private labs. There are 14 private offices. They're all occupied, AV and conference facilities. And I can tell you, there are 35 companies already in the incubator. There are 15 on the waiting list. So this is the hottest commodity in Southern California for startup companies that are coming up with the new therapeutics, devices, and diagnostics in LA County. And the way to keep them here and the way to keep the jobs in LA County and get more jobs in LA County is by having this kind of an incubator. But they have to graduate to the tech park. And so we call it the Bio Center, which is our tech industrial park. And this would resolve a key lack of wet lab space for manufacturing. As I said, there are no manufacturing facilities that these companies can go to. So we have an option for building out 250,000 square feet of buildings on 9.6 acres on the campus. And it'll later grow to 15 acres and up to possibly even 1 million square feet. Now, if you combine Harbor UCLA Medical Center with the LUNDQUIST Institute, with the Biolabs Incubator, with the Bio Center, this will be the first full-service bioscience park in LA, soup to nuts, the bioscience hub, it'll all be there. So our campus master plan looks something like this. This is the part of what used to be Harbor General Hospital, where a new hospital is being built and in the next 5 years will be completed. So the old hospital isn't going to be there. There's going to be a state-of-the-art hospital, about 400 beds, an 8-story outpatient facility. The county is investing $2 billion in upgrading the medical center. Now look where the LUNDQUIST is. It's right next door on its own 11-acre campus, and that's where the incubator is. And lastly, this last piece of property is going to be the Bio Center, the tech park. So just imagine, here you are, you're a physician in the hospital, you see patients with something that you don't like, you go to your lab at the LUNDQUIST, and you come up with a solution. We patent it, becomes intellectual property, and then you go to the incubator where you generate your proof of principle, you attract some venture capital, you need to scale up, you go to the Bio Center, you rent some space there, you have enough space to manufacture what you need to do what? To go back to the hospital where you saw the problem to begin with. So you walked about 50 yards to your lab, you walked another 100 yards to here, you walked 150 yards back, and guess what? You've done it all. You don't have to move, you don't have to ship stuff to Northern California, you don't have to ship stuff to Massachusetts or North Carolina to get your materials made. You can do it all on our campus. And the very last thing in talking about a hub is the fact that people say, you know, this can never happen in LA. LA is too spread out. It can happen in La Jolla. It can happen in San Francisco, but LA is too spread out. Well, that's not true. If you look at, for example, from UCLA to University of California at Irvine, you've got major airports, LAX, Long Beach, John Wayne. You've got academic institutions. You've got biotech companies springing up all over the place. You've got major medical centers. You've got major universities and Cal State colleges. So if you start at UCLA and you go to UC Irvine, who's right in the middle? Us. And so that's the story going forward. We call this the 405 corridor, or the north-south corridor. And you can see that this is a hotbed going forward. And wouldn't it be great to be right in the center with the first bioscience hub in the country and maybe even in the world, right there at the LUNDQUIST. So I want to finish up just giving you one last message. And that is, and this is a pitch, as I mentioned, we are supported largely with federal funding. That's a pass-through. That goes for research, to support research that's ongoing. But we have to innovate. We literally have to be able to have extra money to do the things that we can't get grants to do. New ideas that aren't ripe yet. To produce what we need to do the clinical trials, like I talked about the monoclonal antibody. So I would like to just encourage you, before I open the floor for any questions that you might have, to think about Giving Day. Tomorrow is Tuesday, as you know, it's a national, we call it Giving Day, the week after, the Tuesday after Thanksgiving. And we have a website, LUNDQUIST.org. Very easy. If you can remember LUNDQUIST.org, there's a button there for donations. And we would be very happy if you were able to support the research at the LUNDQUIST. I think you've seen, hopefully, that in one way or another, we've touched all of your lives with some of our discoveries or some of our paradigms. And we will continue to be doing that in the future. And to be able to do that, it goes without saying that we need support beyond just federal grants and other sources of revenue, corporate sponsored research and clinical trials.